Now we will look at transcription and translation, which are the two parts of protein synthesis. So in the process of transcription, we take the information in DNA, in one or more genes, and then transcribe that into a strand of RNA called the RNA transcript. And this is going to be our messenger RNA, or our mRNA. The RNA strand is going to be complementary to the DNA strand. So we have this coding strand of DNA, and the RNA is complementary to that. But remember, in RNA, instead of thymine, we have uracil. So we'll have still adenine, guanine, and cytosine, but this time we're going to have uracil. And it's going to basically be the copy of the gene that contains that information to make the protein. In order for this to happen, we have to get the DNA double helix to partially unwind, just like we did with DNA replication. Instead of a replication bubble, now this area is called the transcription bubble. And so we have what's called the antisense strand, or the coding strand, that will be used for this. Now, in bacteria, when we look at the process of transcription in bacteria, they use RNA polymerase as the enzyme that's going to be used to transcribe all of their genes. And basically, RNA polymerase does the same thing as DNA polymerase. It's going to add nucleotides one by one to the growing nucleotide chain. The difference is, instead of them being DNA nucleotides, now they're going to be RNA nucleotides. And RNA polymerase does not require that primer like we saw that we needed in DNA replication. In transcription, we have the initiation part that begins at what we call a promoter. This is a piece of DNA where the machinery, the transcription machinery, is going to attach. And so this is basically the beginning. The RNA polymerase knows, this is the start point. Then we get elongation, which is just the adding of the nucleotides. And in bacteria, this is about 40 nucleotides per second. And as this elongation process proceeds, the DNA is going to be, unwrapped ahead of it and then wound back up into its helix behind it. And so you've got this RNA polymerase as your enzyme. You've got your two strands of DNA here. Again, we've got the template and the non-template. And the template strand is going to be the one that's running from the 3' to the 5'. And so you'll see we've got this little promoter. Again, that's the trigger point, or the starting point. And we're going to unwrap the DNA, and we're going to build our nucleotides here, our RNA, in this direction. And after we get, this piece built, then this area of the DNA is going to wind back up. And then as we continue on, in this direction, this part of the DNA is going to unwind. Once the entire gene has been transcribed, then the termination must occur. This is when the RNA polymerase dissociates or disconnects from the DNA template, and then it lets go of that piece of RNA that it has made. And this, in the DNA template, we have these pieces of nucleotide sequences that act as these termination signals. In other words, this is the point at which the RNA polymerase can't continue, and that's how it knows it's the end of that particular gene that's going to code for that protein. Now, when we look at transcription in eukaryotes, we're going to see a few differences. There's going to be 3 different forms of polymerase: RNA polymerase 1, 2, and 3. And these are all completely different from the bacterial RNA polymerase. Each is going to transcribe a different subset of genes. But the most important difference is the fact that eukaryotes have a nucleus, whereas prokaryotes don't. So with eukaryotes, this process of transcription is going to have to happen in the nucleus because that's where the DNA is. And so these RNA molecules that are going to be responsible for encoding this protein, they have to be transported out of the nucleus into the cytoplasm, which is where the ribosomes are found. There are several steps that this RNA has to go through so that it doesn't get degraded or it doesn't, fall apart in its journey from the nucleus into the ribosome or over to the ribosome. After the template has been made, so after the transcription process has taken place, now we have to undergo the translation process, which is actually the building of the proteins. This is where we take the mRNA, it's going to be attached to a ribosome, and we're going to bring in amino acids to build that protein chain. And the translation process converts that information that is found in those nucleotides into the amino acids. There are 20 different amino acids, and each amino acid has a specific or one or more specific words, okay, or sequence of nucleotides that code for it, and that's called a codon. And this relationship between that mRNA codon and the corresponding amino acid is called the genetic code. And each one of these is composed of 3 nucleotides. So there are 64 possible combinations that we can have when we look at this genetic code. 61 of those 64 triplet codons code for amino acids, and the other 3 do not. They code for what we call stop codons, which lets the ribosome know that we're at the end of that translation process. There's also another codon called the start codon, but it does code for a particular amino acid, and that's methionine. So that's its start codon. The genetic code is pretty much universal. Pretty much all species share this same genetic code. There are some odd or unusual amino acids that we see in bacteria or archaea that we do not see in eukaryotes. But this graphic here shows this genetic code. I'm going to make my screen a little bit smaller so we can get it all in there. So what we've got here, the first letter in the codon, the second letter, and then the third letter. And so you can see our stop codons here, UAA, UAG, and UGA. So each one of those is it serves as a stop codon. And then AUG codes for methionine, that's our start codon. What you'll notice for most of the rest of the amino acids is some redundancy. For instance, leucine. Leucine, as long as the first letter is C and the second letter is U, that's going to be leucine. So we have redundancy in here, which means that some mistakes can be made that doesn't affect the overall protein. For instance, if the code should be ACU, but instead it's ACA, well, it's still going to code for threonine. So we don't have a mistake in the overall protein, even though we have a mistake in a nucleotide. So this does help prevent some of those mistakes that could cause the protein to be completely misformed because we've put an amino acid in the wrong place. When we look at the process of translation, we're really going to see kind of the same three processes that we saw with transcription. We have initiation, elongation, and termination. And what is going to be required here for this to take place is we have the ribosome, and the ribosome is composed of a large subunit and a small subunit. We have our piece of mRNA here, where we have the AUG start codon at the 5' end and our UAG or whatever other stop codon at the 3' end. And then we've got this other special type of RNA called tRNA. And I like to think of tRNA kind of as a helicopter that's bringing in the amino acids. Each tRNA is going to have what we call the anticodon that is complementary to the codon on the mRNA. That's how it knows what amino acid to bring in. So, for instance, the AUG we know codes for methionine. So the anticodon for that is UHC, that's the complementary base pairing, and it's going to be carrying the amino acid methionine. So it's going to come in, okay, and the large and the small subunit of the ribosome kind of clump together, and the tRNA brings in that first amino acid. That's initiation. So we're going to have methionine. And the ribosome is going to move down towards that 3' end, and we're just going to keep bringing in amino acids one by one with the tRNAs. And again, they're going to have an anticodon that matches up perfectly. It's going to be complementary to those base pairs in the mRNA. Again, that's how it knows what amino acid to bring in. And these tRNAs and these amino acids are just going to kind of be standing nearby. We basically got this pool of amino acids and tRNAs. And as the ribosome reads, that next sequence, it's like, okay, this is what we need. And so the tRNA is gonna bring it in. And as it drops off those amino acids, basically each amino acid is going to kind of form a peptide bond, and we're just going to keep growing this chain and growing this chain until we get to our stop codon. The release factor is going to be here, recognizes that we're at the end. The polypeptide here, our protein, is going to be released, and the ribosome is going to disassemble back into its two subunits. And then the mRNA is going to be recycled into making another of the same protein, or we're just going to snip it all apart and reuse those nucleotides to build some other, mRNA strand. Just to reiterate, some differences here that we see. Again, because prokaryotes do not have a nuclear membrane, everything just takes place right out in the cytoplasm. We get, transcription that takes place, we get translation that takes place, and we, build each one of our little strands or proteins. But in eukaryotes, because we have the nuclear membrane, the RNA is going to be made in the nucleus, and then we have to kind of package it to protect it from being degraded. Then it will exit the nucleus, go out into the cytoplasm, and then that's where the translation process occurs. So in eukaryotes, it all takes place in the cytoplasm. In prokaryotes, it all takes place in the cytoplasm. And in eukaryotes, the first part, transcription, takes place in the nucleus, and then translation takes place in the cytoplasm at the ribosome.